Hello, and welcome to the launch of our Let's Talk Disease webinar series for ornamentals, presented by the Ontario Ministry of Agriculture, Food and Rural Affairs and Greenhouse Canada magazine. So in addition to our guest speaker today, each webinar in this series will be moderated by an OMAFRA greenhouse specialist. Today, we have Dr. Chevonne Carlo, who is the Ontario Greenhouse Floriculture Specialist. In her role, she works to connect growers with the latest information on floriculture crop production. She is also involved in a number of collaborative research and demonstration projects in the sector. Welcome, Chevonne.
Thanks, Greta, and hello, everyone. I'd like to introduce our speaker for today. Our speaker, Dr. John Lea-Cox, is a professor and research extension specialist for the nursery and greenhouse industry at the University of Maryland. His primary expertise is in the areas of water and nutrient management, and his work aims to systematically reduce the environmental impacts of industry production practices. He has a bunch of major research interests, and these are in the areas of wireless sensor networks, soilless substrate, disease management, frost and environmental monitoring, and many more. John's going to talk to us today on the topic of cultural controls for managing and preventing disease. Welcome, John.
Chevonne, thank you very much. I appreciate your introduction and invitation from Ontario. I'm really excited to be here today. So to start off, I wanted to start with a few acknowledgements right up front because disclaimer, first of all, I am not a pathologist, but I have had the benefit of working with a number of very good pathologists over the years, and just to mention Dr. Xuan Hong and Dr. Ping Hong at Virginia Tech down in Hampton Roads, as well as Dr. Gary Moorman, who was at Pennsylvania State University until he retired a few years ago. We worked on a project called the SERI Path project at that point, and I'll show you a little bit of information from that project. And more recently, I've been working with Dr. Cassandra Sweat and Johanna del Castillo, who were at the University of Maryland. Cassandra actually transferred back to UC Davis about 18 months ago. We're still working on the SERI Water 3 project, which I'll tell you much more about. And associated with that project is a PhD student, Ms. Justine Belliot, and my colleague at the University of Maryland, Dr. Andrew Ristley, and also a PhD student in our lab, Mr. Brooke Bolognier. And if it wasn't for these people, I wouldn't be here today. So just also to mention that there are some upcoming webinars from actual pathologists, Dr. Ann Chase, who's going to talk about fusarium basics next week. And then followed by our own Karen Rain, Dr. Karen Rain from the University of Maryland, who's going to talk about disease diagnostics on Tuesday, the April 2nd, as well as Jean Woodward from the University of Georgia in Athens, who's going to talk about powdery mildew control. Anyway, so lots coming up, and anyway, what I'm going to do is try and set the stage from a horticulturalist's perspective, because that's what I am. I'm actually trained as a plant physiologist with my PhD. I've always been primarily associated with nutrients and water management. Those are my two primary interests. I am nursery and greenhouse extension specialist here at the University of Maryland. Mostly on nursery, but greenhouse is a part of the continuum. And so because we work with container crops, substrate selection, site engineering, irrigation, and water management are very much in myself as well as Andrew's purview. So the focus today is really going to try and focus on integrating how we integrate cultural pathogen management practices, because it's not just pathogen management. Obviously, many of you are familiar with the disease triangle, and what we're really trying to do is manage the disease, the environment, and cultural practices to minimize any chance of disease. When we recycle water as well, we obviously are involved with some other remediation techniques, sanitation, chlorination, filtration, and I'll touch briefly on those as well. That's really the focus of the SCRI Water 3 project, and that project is led by Dr. Sarah White at Clemson. That's an ongoing project, and what Cassandra and myself and Andrew have been working on as part of that project is the reduce component. It's called Water 3 because it's reduce, remediate, and recycle, and it's all about water, and we'll get a little bit more into that as I go through this webinar. And then I'll end up with a little bit of work that we've been doing in association with disease management on investigating precision irrigation management, because we're moving towards trying to get to a non-chemical approach to create antagonistic environments for plant growth. And I think we have the capability of doing that with very fine control of irrigation through sensor networks, and I'll spend a little bit of time on that when at the end of the seminar. So just to briefly recap, we work in all environments, not only greenhouse. This is a flood floor, poinsettia, obviously, but we do run the gamut through, certainly here in Maryland, a lot of container growers who are out of doors. They actually cover these hoop houses in winter for winter protection, but during the summer they're open to rainfall, and these growers grow a tremendous variety of crops, and as you can see in this picture, a kind of a mash of crops, which are oftentimes very difficult to manage from not only a water perspective but also a nutrient management perspective. And then we have hybrid operations. This is a pot-and-pot operation where we're actually using precision irrigation on these rather than typically this is an overhead irrigation situation. But when we move into these larger containers, then it's more cost effective for us to use drip or micro sprinkler application of water, and that's usually done through the drip line. So that actually makes things a little bit easier to control in terms of water management. I wanted to just focus in on really on the container, because this is where obviously everything happens, and there's an awful lot going on here in terms of if this is just a sialized plant. We're obviously trying to apply water. We're trying to not over apply water in terms of bleaching nutrients and water out. We find that standing water, obviously, with overhead irrigation and sometimes even mismanaged drip irrigation, can lead to pathogen management issues. So it's a continuum, but that irrigation water application is it's quite difficult because it's very dynamic. And so on a day-to-day basis, we're getting all sorts of influences from the environment, rainfall, temperature, light, obviously, and that all influences the amount of water that plant is using on a daily basis. And so what we're trying to do is replenish that water adequately and most efficiently. And that can become difficult, particularly when we're using overhead irrigation. Of course, we are all faced with, at times, water security issues in terms of actually having enough water to apply. I know you folks up in Ontario have had groundwater issues and nutrient management issues as well over the years. And although you're like us in a relatively water-rich part of the United States and Americas, North America, there are many other regions, of course, and probably many people listening into this seminar who are actually in regions where water is much more critical and much less available, whether it be groundwater and or rainfall. So when you're trying to grow crops under those conditions, water efficiency becomes very important. And that's when we're really getting to the fact where even here in Maryland, we are doing a fair amount of recycling of water. And so that's why it's a bit of a dilemma for nurseries and greenhouses, because once you close the water loop, in terms of recycling water, the potential for unwanted issues in your water returning to your crop become more prevalent. And it becomes even more complicated because some plants obviously are maybe more susceptible to disease or other issues, whereas other crops are less. So sanitation and filtration become very important in those situations. Chevonne and his group have identified a number of isolates of various Phytophthora in irrigation ponds. This is actually a relatively dated list, but Hydropathica and Irrigata, these have varying degrees of virulence. Obviously, if you're talking about Phytophthora cinnamomi and Citropthora, those are extremely aggressive species. But what we're learning from recycled ponds is that there's an awful lot of Phytophthora and other organisms in there that have less virility and don't necessarily lead to disease. So they can be easily controlled with good, effective sanitation and filtration. Of course, you're all familiar with different life cycles of different fungal diseases. This is just one, of course, with waterborne pathogens, zoospores. But we do know that both crop species as well as the zoospores are quite sensitive to stress at different types of their life cycle. So what Chevonne's project did very nicely was actually explore some of the work that how salinity and somehow the other temperature and other factors in the pond can actually affect zoospore longevity. And I think one of the main things that Chevonne, out of Chevonne's project was that, quite frankly, make sure that your pump inlet was placed at a not only an appropriate depth, so it's not sucking up material from the bottom of your pond, but also is away from your inlet. So you're increasing that travel time that zoospores might have if they are going to be transmitted back to the crop. Then you're trying to lengthen that path. So there's lots of different ways that disease can be managed in the nursery. But I wanted to just, before I kind of move on to some specifics, also mention Jennifer Park's group in Oregon State. Has done some fantastic work over the years. And in fact, Jennifer is also part of the SERI Water 3 project. And what Jennifer has done, and in fact might be even a suitable topic for a future webinar, really looked at hazard analysis and looked at the critical control points that you can affect and ensure that those conditions in the nursery or in the greenhouse are taken care of. And that's what I'm going to focus on next. Some hazards are, and some sources of inoculum are much more important than others, and of course it's those which we need to take care of. So when we're thinking about disease management, I want you to really think about, maybe for your own operation, how your system is working, because as part of the SERI Water 3 project, what we're really trying to do is not only look at pathogen management, but we're also trying to look at nutrient as well as some of the other herbicide and even growth regulator runoff issues. So this treatment train approach, where we're using non-chemical approaches to reduce the overall level of contaminants running off that can be then more adequately filtered and recycled back to the crop, that's the point of the project. So, some integrated pathogen management practices. And for those of you who've been through any good course in pathogen management or pathology 101, this is probably not a surprise, but of course there are really a number of things that you really should focus on. And I'll go through these in a little bit more detail in the next few slides. Firstly, clean stock. clean plants, but also substrate formulation as well as source is very important in terms of providing an adequate environment or an antagonistic environment for pathogens. I'm going to get into a little bit of good site design in terms of irrigation pond management and actually show you some resources that we have online for that. And then I'll get into some of the precision water management techniques that we're working with. More importantly, it's being able to actually apply this knowledge in a systematic way that is really the most important point to take away. So clean stock. Obviously, propagating material. If you're importing nursery stock from other nurseries, other areas, there's always the chance of you bringing in other unwanted pathogens. And so even just an initial screening, or if you suspect that any stock is actually contaminated, ensuring that you take advantage of the services of somebody like Karen Rain, who runs the disease diagnostic clinic here at the University of Maryland. Those facilities and Karen for us is an incredibly important part of us being able to ensure that we are maintaining clean stock, not only going out but also coming in. Of course, growing media and clean production areas. If for any of you who are in production or oftentimes substrates are not on concrete surfaces; they're often on dirt surfaces. Water may run off from the nursery into those areas, and so there's an awful lot just in ensuring that any media that you have is clean up until the point that it's actually used. Many of our nurseries actually use composting, may have composting facilities on their property, and we try to ensure that those composting facilities are well away from any production areas. They're also contained and even screened because a lot of this material can be windborne as well. So just be aware that disease can not only come from water, it obviously can come from wind. And quite honestly, weed management and windborne weed seed is probably more an issue for keeping your substrate clean in that particular with that particular factor. Also be aware of volunteer host carriers, because there are many other, maybe even native species, certainly ornamental species that could be hosts of various and basically asymptomatic hosts. So they look perfectly healthy, but they are carriers and they may be an inoculum source. So you want to make sure that those are certainly kept away from production areas. Substrate formulation is extremely important. It's probably Andrew Ristby is our substrate expert here at the University of Maryland, and it's amazing how many substrates are poorly formulated in that they hold too much water, and they tend to be, and combined with over irrigation, they really don't have adequate aeration. And of course, good root growth is dependent on good aeration as well as enough water and nutrients. So it's a kind of a fine balance between airfill porosity, water holding capacity, and good drainage to ensure that you don't get inoculum building up in the substrates over time. Production areas, of course, this is oftentimes the place where we really do see issues arising, particularly where production areas are saturated. It's very difficult, and I'll get into a little bit about irrigation efficiency in a bit, because irrigation efficiency oftentimes is the resulting cause of production areas having too much water. So there are oftentimes low spots that actually where water ponds that become sites for pathogens to thrive and spread. So when we're looking at conveyance systems, good area production design is really about moving water off the production area bed as fast as possible, and also just providing that drier environment to be able to ensure that if there is any runoff, particularly with stormwater or normal rainfall, that those production areas don't get saturated. In many of our container nurseries, we typically have weed barrier on those container production areas, usually sitting on top of gravel. So water, particularly with overhead irrigation, as you can see on the right-hand side of this slide, in order to get enough water into those containers, very often there's an awful lot of water going on to that crop. And if that is removed from the production area and moved back to a recycling pond efficiently, that can be a good way to resolve any water management issues. Recycling pond design, as I mentioned with talking about Chevonne's project, pond design is extremely important. And one of the other things we learned from his project is if you have a 2 or 3 pond design where you've got the first pond that is really capturing your sediment load and is easily, even if it's a small area, that can actually just drop your sediment load and actually be easily cleaned out before your main pond, that goes a long way to actually dropping out a lot of not only sediment but also zoospores. And then if you have another pond, which is basically after the second pond that you irrigate from, that provides another step in terms of being able to create that treatment train effect. So we have a knowledge center, which I just wanted to throw up now before I forget about it. This is a free site you can go to. It's called WaterNut, water nutrients, waternut.org. And if you put a /Moodle, Moodle is just the learning center, the learning management system. But if you put that URL into your browser, you'll be able to actually go to this site, and we have a number of learning modules on this site, one of which is Containment Basin Design. Some of these modules are password protected just to keep the spammers and the bots out, but you can easily just provide your details, log in, create your own password, and it'll just do a quick security check, and it will you just click on that link and it'll get right in. So what we have in those learning modules is literally a step through on literally what is good containment basin design, and this was developed by a colleague of mine, Dr. David Ross, a number of years ago, but it gives a very nice walkthrough of exactly what it takes to create a very good containment basin design in terms of not only sediment control but also pathogen control. This is one site that one actual case study that we highlight, and here you can see that they've got actually the inlet, pond inlet goes around the outside of the main pond. And they did it this way because they didn't have enough space to create that second pond. So they created this narrow, long path bed with riprap to drop their sediment. And this provides a nice long path for that to do that slowly. Any water coming in off their production areas slowly returns to the pond, right to the top of this pond at the top end there. And then this is a good way to affect that good water quality before it enters your main pond. So there's lots of things that affect pond quality. Obviously nutrients, returning phosphorus. That's a subject for another day. But recognize pond water quality is oftentimes affected by, if you've got poor practices in your production area and poor design and maintenance, you're always going to have issues. So it very much is a systematic endeavor for a production system. So moving on to irrigation management, because this is where we really, I think, have a remarkable opportunity to perhaps provide an antagonistic environment for pathogens. Very often, irrigation systems are initially adequately designed, but over time, they can become, they're not necessarily well maintained. They could be extended and then suffer from low flow issues, or so there's lots of things that can affect irrigation and irrigation management system over time. And it's very important. Irrigation efficiency is absolutely tied to irrigation maintenance. Without correct pressure, you're not going to get adequate coverage, and we've all seen it. Unfortunately, irrigation maintenance, there never seems to be enough time to maintain sprinkler heads and make sure that they're not worn, or obviously in the right-hand side, that's an extreme case that I'm sure does get fixed, but irrigation sprinkler maintenance is sometimes things, it just doesn't get done. And that affects your irrigation efficiency. That means that plants in the drier areas have to get irrigated for a certain length of time, and usually it's probably 5 or 10% of the area that doesn't get adequately irrigated, and then 95 or 90% of the area that gets over irrigated. So the problem becomes that with those kinds of conditions, you're always going to have standing water, you're always going to be using too much water, and that's when we often see pathogen management issues coming to the fore as well. Again, there's actually another module in there. It's called Irrigation System Audits. So there is a very nice learning management module which will actually run you through and show you exactly how to do an audit of your irrigation system to be able to get it up to good efficiency. So irrigation scheduling, unfortunately, is oftentimes not a terribly thoughtful process. We're all very busy, and unfortunately, a time clock is not an intelligent way to irrigate. Obviously, irrigation managers are integrating an awful lot of information on a daily basis, whether it rained yesterday, whether it's very hot or windy today, and so they're always constantly battling how to manage that time clock. And so it's a very difficult issue, particularly when you're growing many different types of crops. If you're in a greenhouse environment, it's probably less of an issue because you have higher efficiency systems, you've got booms, you have much higher densities, you don't necessarily have the issues with you have a controlled environment. So there are issues that are more prevalent in the nursery industry, but even in greenhouse situations, irrigation is oftentimes one of those things that could be improved. Also, I think what we're trying to do is investigate pathogen management by using the sensors to understand when to apply water, because oftentimes it's with the high use of fungicides as a preventative, we're often masking the symptoms that we often see when our plants are under stress. So our central question in this endeavor is, if we can irrigate just based on plant needs, and we can keep the plant relatively stress-free, does that antagonize pathogen populations in the root zone? And could that possibly reduce the need for fungicide use? So with some of the work that I'd done, I led this previous project called the SCRI Mines project. We spent a lot of time looking at ways to control irrigation, and it's done through sensors and sensor networks. I won't get into too much of that today, but basically just to show you that it involves just having a piece of hardware. It could be what we call a node that is connected to some sensors, and it's those sensors that actually drive the decision that the application of water is controlled by. So this node on the right-hand side, you probably can't see it, but so on the right-hand side here, there's a solenoid valve. And so what happens is those sensors are placed in the crop, and with a particular threshold volumetric water content, that turns the water on and off to be able to only irrigate when the plant actually needs it. Now, obviously, that's easier when crops are irrigated with precision irrigation techniques like drip or micro sprinkler. But we have found, even with overhead irrigation systems, that we can actually cut down the duration of irrigations by almost 50%. Depends on the crop, but usually between 40 and 60%. That's the reduction in water that we're typically getting on average. And the reason that we can achieve those savings is we're oftentimes, as irrigators, very tuned into starting the irrigation, and we know when the crop needs water, but what we don't really know is when to turn the irrigation off. And because we are conservative and risk averse, we tend to overestimate the amount of time that a crop actually needs. So it's just literally by understanding and having that information to know when to switch the irrigation switch off, irrigation control valve off, that's where we achieve most of our savings. We have software that really helps do this. It's I won't get into this if you have any questions, I'm happy to answer them at the end. But we have an ability to actually apply very short durations, what we call micropulse irrigations to containers, where we can literally apply 5 or 10 seconds of water, allow the sensors to have a timeout and to measure that water for, say, 5 minutes later. And if it needs water again, then it'll give it another micropulse. So that way, what we're really doing is just gradually topping up the container with water and not spending an awful lot of time over irrigating it. Matthew Chappell, as part of the Mines project, did some work on Gardenia, and what was very interesting, Gardenia is a very problematic crop for this one nursery down in Georgia, and what was very interesting was that they enabled the nursery to actually reduce their crop loss to Phytophthora to virtually zero, just by manipulating the irrigation schedules. What was more interesting from that particular paper was that they also reduced the production time of that crop. So because it was less stressed, this was all compound interest, and so they produced a lot more of that crop literally in a shorter production period. It's a little bit complicated because it went over a winter period, so they actually managed to get that whole crop produced in 6 to 8 months, which meant that it actually they turned that land over. Instead of taking 18 months, they turned that land over in 12 months or less. So there was a real economic gain in terms of producing that crop with reduced amounts of water because of the disease. If you're interested, you can go to that website. The project website is smartfarms.net, and there's a whole host of papers and impacts of that project that you could explore. Just to finish up, because I'd like to finish up in the next 5 or 10 minutes so we have adequate time for questions. We've been moving on with from the Mines project to do some larger scale commercial work with sensors with various operations. We're working with 2 actually in Maryland, one of which is a large greenhouse operation, and they chrysanthemum and poinsettia are 2 of their major crops that they produce. And so what we've done in some studies a couple of years ago, we were looking at their outdoor production areas, looking at what they were doing in terms of irrigation control, excuse me, and what we could do with sensor-based control. So with chrysanthemums, these are not your normal small size chrysanthemums. These can be quite large, and so this is Brook Bologna in that chrysanthemum field. These are all on drip irrigation, and so they take about 8 to 12 weeks to produce, and they're beautiful plants. But as you can imagine, they use they can go through water very rapidly on a daily basis. And so just to give you an idea, this is, I'll spend a little bit of time on this. This is the blue line was our sensor-based control. The black line was the actual average of the sensors with the grower control. And if you can see on the right-hand side, there's the volumetric water content, 10 up to 50%. So we were maintaining our set points on these chrysanthemums at about 40%, thereabouts, on a daily basis, perhaps going up to about 45%. Whereas the grower control, because of scheduling and because these were mostly irrigated at night, during the day, those volumetric water contents in the containers dropped to sometimes around about 10% water, which we were extremely initially quite concerned about, because we thought that, quite frankly, it was going to translate into a large reduction in growth. But what turned out is in fact there was very little difference. We actually saved about 10% of the water that we were applying compared to the grower. But what was really curious when we did the analysis of the plants, they had very little significant difference between what we were applying and what the grower was applying. So this is a salient lesson, because I think what it goes to show is that as long as water is applied within a particular time period, and as long as that plant doesn't get into a serious water stress, it is probably okay. So again, it's not the overall making sure that the water is maintained at a particular constant rate. As long as you replenish that water within a certain period and you don't let that plant get into severe stress, it probably won't translate into growth differences. Just to move on, just a few last results from, this is Johanna Castillo, who is doing her postdoc work at Maryland. Johanna has been working with both tomato as a kind of a standardized crop, but also with poinsettia. So just to very briefly show you some of her tomato work, literally the outcome of the tomato. Very similarly, what Johanna did, which we couldn't do at the nursery, is we couldn't inoculate. So what Johanna has been doing with her studies is she inoculated tomato with Phytophthora capsicum, sorry, excuse me. And then we looked at the difference between inoculated and non-inoculated plants, both tomato and poinsettia. And what we were looking at is trying to find out whether if we grew them at a lower irrigation volumetric water content, not that they would get stressed, but that we could dry out that root zone, whether that would have a substantial effect on, firstly, the inoculum and also the plant quality. So this particular study was done in sand, and that is why the volumetric water contents are low. But what you can see is between the normal 20% volumetric water content, which was our control, and the 15, there was very little difference. Whereas when we got to a very dry volumetric water content of 10%, that was when we did see an increased disease severity with tomato. So with poinsettia, this paper has actually just come out. I think it's just in press. So with poinsettia, we had somewhat similar results in that they are grown. This is grown in a peat-based substrate. So that's why the volumetric water contents are higher, but literally between 45 and 35, it doesn't sound like a lot in terms of water content, but 45 is virtually saturated, whereas 35 is a lot drier in terms of overall media water content, whereas 25 is very dry. So the point being is that 45, if you reduce your water content, reduce that leaching fraction, create a drier environment, you actually don't get any effect on disease severity. But when you do get to a lower water content, that's when you could bump up the amount of inoculum that could be affecting your plants. So just in summary, to sum all of this up, it seems like we can reduce water applications without significantly affecting plant growth. Of course, we've only done work with a few species, and there may be many crops that that doesn't apply to. But I think we're showing that we can actually reduce the amount of water, not affect plant growth, and maybe we can actually reduce the amount of inoculum that is spreading, has the potential to spread. Most importantly, though, what I wanted to really you to take away from this webinar is that you need to take that treatment train approach, look at that critical control point analysis that Jennifer Park has elegantly outlined, and really understand your system in terms of clean plants, clean substrate, reducing your irrigation, improving your irrigation efficiency, reducing your water runoff, and integrating all of the moving parts of the system to by and large affect disease. What we have shown is that when you do that, the chances of pathogens returning to the crop have been shown to be reduced very significantly, and particularly when filtration and chlorination or other sanitation systems are employed. And with that, I will just show you a few last screenshots. The WaterNut.org Knowledge Center, which you're welcome to browse through, as well as the SCRI Path website. This is Chevonne Hong's project. That they have many resources and papers. Please explore that as well. And my thanks to my colleagues in the Clean Water 3 project. That project is just finishing up this year, but there's lots of really nice information from various parts of that project. It's a very good and wide-ranging project, which I hope can can help you with your information needs. Thank you, Greta. Thank you. Thanks, Chevonne. If anybody has any questions, I'll happy to take them.
Awesome. Thanks so much, John. Really well-informed presentation. You covered a lot of topics. Thanks so much. We do have a couple questions. So I'll just start by reading them off and maybe give a little context and then toss it over to you to sort of answer. So the first one, if plant tissue is already infected by soil-borne diseases like Phytophthora, can plant growing under proper climate management techniques isolate or stop the growing pathogen? You sort of touched on this a little bit with the poinsettia slides, but it sort of seems like a lot of articles out there indicate that an infected plant is more like a dead plant or might just equal a dead plant. But some growers would say that under controlled environment situations, it seems like they might get results along a spectrum of dead plant to stunted plant to perhaps a plant that might be marketable on some level. Can you speak to that question?
Well, I'll try, but I'll firstly put out that I'm not a pathologist, so that would be a really good question to ask Karen Rain or one of the other pathologists in the next couple of weeks. I'm going to go back to some of my own personal experience. Right after undergrad, I started, I was a citrus nursery manager in South Africa, and I was straight out of college and I thought I knew everything. And then unfortunately, I walked into the biggest Phytophthora situation in that nursery that you could imagine. And we spent an awful lot of time trying to clean up that nursery. Eventually, we employed the services of a very good plant pathologist, consultative pathologist. But what we had to do is we had to go right back to basics. And we had to do all of the things that I basically talked about, because it was clear that we could throw as much chemicals as we could at that problem, but we were never going to solve the issue. I personally think that the only way you can really get rid of a disease management issue is by going back to basics and, quite frankly, roguing out all of that suspected disease material. And I've never seen disease material. You know, I think it's a gray area. I think there's probably always some disease in root systems. I'd like to hear an expert actually speak to that. But I think if you've got an endemic problem, you've really got to go back to basics. And it's very hard to clean up a disease management issue without doing that.
Right. Yeah. No, that's a very good point. Okay, getting back to those basic parts, we've got a question on your thoughts around reusing containers and sterilizing them for reuse. So I'm interpreting a little bit here from the question, but I assume the containers would be for plant pots or mixing things.
Well, I think a lot of growers do this, and of course, containers are extremely expensive, and so it's always let's say it's always a temptation to reuse containers, but unless your cleaning protocols are exceptionally good, I think there's always a risk of you recontaminating a crop with a dirty pot. However, I think if you've got good, if you've got a good system where you can chlorinate, you can use sodium hypochlorite, for example, to clean those containers, and you're really scrubbing them out, you could do it. Unfortunately, I hate to say it, is the cost of a new pot compared to the labor it takes to clean a pot is probably the most important factor for most operations. And so, the risk is always there by reusing pots, I think. Again,
Right.
ask the pathologist.
Good point. Yeah, for sure. The effort that you would put into sanitation versus the cost of a new pot is a good analysis to sort of look at for your own operation.
I wish we had more pot recycling facilities than we do.
Yes, yeah, for sure. Okay, moving along. Does the amount of fertilizer applied when the plants are watered, do you think that that has an influence on the risk of disease?
Good question. That's a very interesting question, because I don't have any direct evidence that it does, but I would certainly say that if you are, and I would say particularly with some of the micronutrients, and perhaps not deficiencies, but perhaps when you have got some micronutrients in toxicity, I'm thinking particularly with pine barks and manganese, you could be stressing roots in that many pathogens, I as far as I'm aware, are opportunists, and even the more aggressive pathogens oftentimes need a wound or some kind of root stress to gain entry. A healthy root system, it's a bit like our own immune system, at least that's my simple analogy, is that when you're healthy, you've got a good resistance. But so I think what's interesting is, I think if you over apply, particularly nitrogen and phosphorus, you will find that your root to shoot ratio will become high. That means you'll get a large shoot for a small amount of root. Now the danger is, of course, when you've got that and you're trying to manage irrigation, is that the plant is pretty sensible in that it doesn't put carbon into a root system if it doesn't need it. But the fact is, when that plant suddenly gets into stress, and if it's getting into water stress, it's losing water out of its leaves, it doesn't have enough root to supply, that's when you can get into stress, and that's when I think you could get into a situation where pathogens could gain entry.
Makes sense. It sort of goes back to that sensitive to stress thing that you were talking about at the very beginning.
Yeah, and different plants have different root systems as well. So, if you've got a plant with a very fibrous root system, like a holly, compared to an azalea with which has got a very fine root system, I think fine root systems probably have a greater propensity to be affected by stress. So, again, that's when, quite frankly, I defer to the growers. Very often they know what their problem kids are in terms of stress and indicator plants, and so that's where I really defer to them and their knowledge.
Right. Good. Okay, a couple questions on pulse irrigation. So a lot of growers use clear water to leach at particular times to adjust the EC in pots and for leaching of accumulated salts. When you move to a reduction in watering with pulse irrigation, the question is, do we need to add more water for leaching, or would leaching work with a pulse irrigation system?
That's a very good question, and it gets to I think the irrigation system itself, and I'm going to be cautious here, because what I've learned is pulse irrigation works very well with precision irrigation systems. So if you've got a drip or a micro sprinkler, or if you've got a dense, a very tightly packed area, for example, in a greenhouse, when you've got overhead boom, a quick pulse irrigation oftentimes is a very effective way to irrigate, because you're not leaching, and then you can quickly go over. But of course, as the question is alluding to, if you don't run clear water, or don't have a leaching fraction that actually gets those excess salts out of your root profile occasionally, then you could build up salinity. But I think it depends on the precision. So with pulse, you have the ability to pulse it maybe 2 or 3 times. I've seen in some container nurseries, those pot and pot operations, sometimes they'll even pulse 4 or 5 times in a day. But at the end of the day, if they do an irrigation and it's a pulse, it will actually leach out, because that's when the pot is, I beg your pardon, in the beginning of the day, it'll leach out because that's when the pot is most saturated. So it depends, and typically what we do is we actually put an EC sensor in the base of the pot. So we actually have an EC reading as well as a volumetric water content reading. And that way we can manage salts and water in an efficient way. I hope
Great.
that
Yeah,
answers.
no.
Not a very clear answer, I'm afraid.
I'm going to push back on this one a bit, because I have an interest here. So you talked a little bit about how greenhouses are lucky, or we should consider ourselves lucky because we have more control than a field grower would. Definitely that's true, but I know I've seen a lot of growers struggle through dark and bad periods of weather in the spring and fall when it's gray outside, there's a lot of rain, and they really struggle to get the crop to dry down or to get proper nutrition into the crop. And we often see that this contributes to disease outbreaks in the greenhouse. So we know that further control, like those EC meters, different things like that, more sensors, you talked a little bit about that, would be helpful. Are you aware of any potential technology solutions that could help with this, that might be coming on board in the future?
Well, that and that low light issue is something that we sometimes experience, and believe me, here in Maryland, even in the last year or so, we've been so wet, and the conditions have been so kind of gray and gloomy that we've experienced those conditions that you would more oftentimes experience in Canada. So it's a very difficult situation. In fact, one of the producers that we worked with on the Mines project was a Snapdragon cut flower producer. And so he was always interested, because his problem was he wasn't getting enough nutrients onto the crop at that particular time period. What I think the key to this is it may take and that having that very wet substrate is a problem, and I know it tends to be more of a problem with the peat-based substrates. Peat is nice because it actually has that internal porosity, which you sometimes don't have with the wood fiber or the other coconut fiber coir. But even so, I think what we could be doing at that point is maybe understanding the substrate irrigation plant kind of triangle, because obviously light is driving water use, but the plant is not using that much water, so we still have to fertigate, and that's where it becomes a problem, because that's when it becomes very wet. So again, going back to the system, is if you can at least keep your production areas dry, even if you do irrigate and over irrigate, which you have to do occasionally, that's probably the key to reducing your pathogen load. But again, ask a pathologist, because I'm talking as a horticulturalist.
Right, yeah, for sure. And I can't comment on that either, because I'm also a plant physiologist. So we had some good questions today, I think. Thanks very much again, Jane.
Thank you, John, for a wonderful presentation, and thank you, Chevonne, for moderating such a great discussion that we had today. For the audience, thank you for joining us. We will have this webinar available on the website, and a link will be sent out to you within 24 hours as well. For more great content on greenhouse ornamental production, visit onfloriculture.wordpress.com. That's a blog that's regularly updated by our OMAFRA greenhouse floriculture specialists. And of course, check out greenhousecanada.com for news and production tips, and sign up for monthly copies of our magazine in print and online. And as John mentioned, be sure to check out our second webinar in the series next week on March 20th. We'll be talking Fusarium Basics with Dr. Ann Chase of Chase Agricultural Consulting, moderated by Greenhouse Floriculture IPM Specialist, Dr. Sarah Jandrasik. See you next time.